Okay, so welcome to this video. In this video, what we're going to discuss is protein folding, okay, and then protein folding diseases. Okay, so let me outline the structure then for this video. What we're going to start off with is a discussion of the normal proteostasis mechanisms. Okay, so we're going to look at chaperones and also the normal degradation pathways for misfolded proteins. Okay, then what we will move on to is the study of examples of diseases which result from proteins misfolding. Okay, and we'll look at broadly speaking, two major classes of protein folding diseases. Okay, so there are the protein folding diseases which result because of loss of function of the protein that misfolds. Okay, so the disease phenotype results because when the protein misfolds, it loses its function. Okay, and it's the loss of function of that protein that then causes the disease phenotype. Okay, so the examples that we'll see there are lysosomal storage disorders. Okay, specifically we'll look at Fabry disease, and the other major example is cystic fibrosis. The major mutation of cystic fibrosis, delta F508, is a folding disorder of the cystic fibrosis transmembrane conductance regulator. So we'll look at that. Okay, then we'll go on to protein folding diseases which are gain of function protein folding diseases, and what that means is that the disease phenotype results because of the protein misfolding, and that misfolded protein now has a new function. Specifically, it's going to aggregate. Okay, so we'll look at the amyloidoses. So we'll look at primary systemic amyloidosis. We'll look at the Transthyretin amyloidoses, so familial amyloid polyneuropathy, okay, and others. And then we'll look at dialysis-related amyloidosis as well. Okay, then we'll move on to Alzheimer's disease, which is a neurodegenerative disease in the brain that results because of amyloid fibril formation. Okay, and we'll have a look at that. Then what we'll move on to is looking at type 2 diabetes mellitus, the potential involvement of amyloidosis in type 2 diabetes mellitus, particularly in the beta cell dysfunction that's seen within type 2 diabetes mellitus. Okay, and then finally we'll move on to the prion diseases, and then finally after that, we'll move on to the serpinopathies. Okay, and the reason I've left the serpinopathies right until the end is that serpinopathies really, the serpinopathy that we're going to look at is alpha-1 antitrypsin deficiency, and that has an element of both gain of function and loss of function. Okay, so the lack of the normal functional alpha-1 antitrypsin causes part of the disease phenotype, and then the gain of function of the misfolded alpha-1 antitrypsin causes another disease phenotype. Okay, so we'll leave that one right until the end. Okay, right. So the first thing then that we're going to discuss is the mechanisms of normal proteostasis. So at first what I'm going to do is just outline the whole of proteostasis. And then what we'll do is we will look at certain portions of this in much more detail. Okay, so firstly then, what actually do I mean by protein folding? Okay, we'll come to what I mean by proteostasis later on. For now, what is meant by protein folding? Well, protein folding means the process by which the nascent polypeptide assumes its three-dimensional structure. Okay, so let me draw a picture for this. So when the polypeptide is first synthesized by the ribosome, it is just a polymer of amino acids. So it literally is just amino acid after amino acid after amino acid. This is what is meant by the nascent polypeptide, the polymer of amino acids that has not yet folded into a three-dimensional structure. Okay, folding is the process by which this now adopts a three-dimensional structure rather than just being a linear polymer of amino acids. Okay, so folding involves it acquiring both secondary protein structures and also tertiary protein structures, and if they exist, also the quaternary protein structures. Okay, so what protein folding therefore involves is it folding into its three-dimensional shape. So maybe it might fold into something that looks like this. So there's an alpha helix, there's some beta sheets, something like that maybe. Okay, and once the protein has folded into its normal three-dimensional state, this is known now as the native protein. Okay, so this is the native protein once it's in that final state. And this process of the nascent polypeptide folding into its three-dimensional native protein structure here, that is the process of protein folding, and it is this that we are going to be extremely interested in this video. This is what we're studying, basically, this phenomenon that proteins go from just being this polypeptide, which is this polymer of amino acids, to having this three-dimensional native structure. Okay, right. Now, certain proteins undergo this folding on their own, okay? It's a spontaneous process. They will, if you just leave them on their own, they will fold into this three-dimensional native structure, okay? Those proteins which do that spontaneously are generally very, very small polypeptides, okay? So not many amino acids, basically. So small proteins can fold spontaneously. Larger proteins need help to actually fold, okay? And the help that is provided within the cell is in the form of loads and loads of proteins, and there is a massive great class of proteins that are involved in helping other proteins to fold. And the broad name for all the proteins within a cell that are involved in helping proteins to fold into their native three-dimensional structure is chaperones. Okay, so this is the name for proteins that are involved in helping polypeptides to fold into their native three-dimensional structure, chaperones. Okay, right. So let's now update our little flow diagram here and put in some additional structures. So let's say that our nascent polypeptide is too long to just fold spontaneously. Then it's likely that what's going to have to happen is the nascent polypeptide is firstly going to form what's known as a folding intermediate. Okay, and this is where the nascent polypeptide has bound with some sort of chaperone protein which is going to help it to fold. Okay, so it's going to make a folding intermediate, which is where the polypeptide is bound to some chaperone in some sort of way. Okay, and we'll look at examples of chaperone proteins later on. Okay, so this involved the chaperone binding with the nascent polypeptide. Okay, and then finally what will happen is the chaperone will come off once the protein has finally folded, okay, and then you'll finally go into the native protein state here. Okay, so now here's the native protein. Now, of course, native proteins don't necessarily remain in the three-dimensional state all the time. They can unfold, basically. Bonds that are holding this native protein structure together can break and the protein can adopt a new conformation, okay? And that new protein, where it's still folded up, but it's now not in this native folding here. It's not in the perfect folding for the protein. It's got a different sort of folding. That's what's known as a misfolded protein. So it can go to become a misfolded protein. Now, of course, just as it can go from being in the native state to being in the misfolded state, it can also go back again. Okay, so this process of the native protein going to being a misfolded protein is known as misfolding. The process of the misfolded protein going back to being a native protein is then called refolding. And certain proteins can refold once they have misfolded spontaneously. Okay, so they can misfold spontaneously and then they can refold spontaneously. However, many misfolded proteins will then bind with chaperones again. They'll become a folding intermediate and then they will go back to being the native protein. Okay, so again, what's going to happen is a chaperone is going to bind to the misfolded protein and it's going to help it to fold back into the native state. And again, I'll say it again, we're going to look at these chaperone proteins in a lot more detail later on. At the moment, I'm just outlining the basics of protein folding. Okay, right. So, why are misfolded proteins so dangerous? Well, basically, the native protein state is the most stable one. Okay, now, when proteins misfold, what tends to happen is they end up not being as stable as the native protein. In fact, that's always the case. They end up not being as stable as the native protein. Okay, and specifically, one of the characteristics that misfolded proteins have, which makes them unstable, is they generally have exposed hydrophobic residues. Okay, so let me explain this in a little bit more. Basically, if we have a native protein, so if I draw my native protein again here, okay, the one that I drew earlier, here it is again, okay, this will have, oops, I've labelled up the N and the C terminus the wrong way around compared to that picture, but never mind, okay, this will be folded in such a way that hydrophobic residues will be buried away in the depths of the protein so that they are not exposed on the surface of the protein, okay, specifically so that they're not exposed to water molecules. Okay, so let me just make sure that everyone understands what I mean by a hydrophobic residue. Okay, so a hydrophobic residue is one with an extremely neutral structure and therefore one that doesn't interact well with water. Okay, so let me give you an example of an extremely hydrophobic residue. So one of the classic examples of an extremely hydrophobic residue would be the amino acid leucine. Okay, so let me just draw the structure of the amino acid leucine out for you. Okay, so I'll draw it as an amino acid residue, which means that I'm going to draw it as though it's within a polypeptide rather than drawing the pure amino acid out. So here's the amino group bound to the carboxylic acid group of the amino acid prior to it. Here's the alpha carbon with a hydrogen coming off here. Here's the carboxylic acid group, again bound to the amino group of the amino acid after it. And now the R group of a leucine residue consists firstly of a methylene group, then with another carbon which will have two methyl groups coming off it, one, and here's the second one, and then also a hydrogen coming off it. So that is a leucine residue, and this is a classic example of a hydrophobic amino acid residue. Now, why is it hydrophobic? Well, all of these bonds in this R group here of leucine, those are very unpolar bonds, okay? So the electrons in those bonds are sitting pretty much equally in between the two components. Now this is the opposite to water molecules. If I draw a water molecule here, okay, water is an extremely polar molecule because oxygen has a much greater electronegativity than hydrogen atoms, and all the word electronegativity means is how hard a atom pulls electrons to it, towards it. Okay, oxygen we say has a much higher electronegativity than hydrogen. That means that oxygen's ability to pull the electrons in this bond here towards it is better than the hydrogen's ability to do that. Okay, so the electrons will sit slanted towards the oxygen atom. And that's for both of these bonds. And that will result in the oxygen atom, therefore, having a partial negative charge. Okay? In addition, the hydrogen atoms have lost those electrons. The electrons are sitting away from them. Okay, so they're going to end up with partial positive charges here. Okay, so that means that even though the entire water molecule is not charged, there is no net charge on the water molecule, the molecule does have an uneven distribution of charge. It is polar. It has a negative pole and a positively charged pole down here. Okay, that is the exact opposite as what we have here to what we have here. Okay, these bonds here that we have loads of bonds in this R group that are between two carbon atoms. Now carbon obviously has the same electronegativity as carbon, so that means these electrons in these bonds between two carbon atoms are going to be completely sitting in the middle of the two carbons, and therefore you're not going to have an uneven distribution of charge. Okay, now what we have to consider is the bonds between the carbon and the hydrogen atoms. Well, carbon and hydrogen roughly have the same electronegativities. Okay, so again the electrons sit roughly in between the carbon and the hydrogen atoms. So again there's roughly no uneven distribution of charge in this R group, and everything is just neutral. Okay, now that means that water molecules cannot interact very well with this leucine's R group here. Okay, because it is so neutral, the water molecules aren't actually going to want to interact with it because they can interact far better with other water molecules. Okay, so if I draw another water molecule here, okay, this one will also have a partial negative charge at its oxygen and partial positive charges at its hydrogens. And what you can actually form here is a hydrogen bond between the hydrogen atom of one water molecule and the oxygen atom of the other water molecule. Okay, so remember, to form hydrogen bonds, what you need is a hydrogen bond donor, which is the hydrogen here. Okay, so this is the hydrogen bond donor here. And the characteristics of a hydrogen bond donor are that it's a hydrogen atom that's partially positively charged. And you also need a hydrogen bond acceptor. And the characteristics of a hydrogen bond acceptor are that it needs to be an electronegative, partially negatively charged atom, which also happens to have a lone pair of electrons. Now, oxygen in water has 2 lone pairs of electrons. It actually only needs 1 to form a hydrogen bond. So it certainly fulfills both of those criteria. So what can basically happen is the partial positively charged hydrogen can form an interaction driven by electrostatics between the partially positively charged hydrogen here and the lone pair of electrons on this oxygen atom here. Okay, and that interaction that we have there is known as a hydrogen bond, and it's a very strong form of intermolecular bonding that can form between 2 water molecules. Okay, right, so water molecules can bind to each other incredibly well. In contrast, water molecules cannot bind to this R group at all well. Okay, so water molecules would much rather bind with other water molecules than bind with this hydrophobic residue. So what tends to happen is when you put polypeptides with hydrophobic residues in water like this, they tend to push all hydrophobic residues together, okay, so that the water molecules don't have to interact with them and they can instead interact with other water molecules, okay. So normally in native proteins what you end up with is all the hydrophobic residues in the center shielded away from the water, and then you have more polar residues on the outside which will be interacting with the water. Now in misfolded proteins what tends to happen is you get hydrophobic residues on the surface, okay, and this makes the misfolded protein what's known as sticky in quotation marks. Okay, so the misfolded protein gets hydrophobic residues on its surface, and this leads to it becoming sticky. Okay, so I'm just going to draw a picture of my misfolded protein. So how am I going to do this? Might just have it, maybe it's now folded up something like that. Okay, so it's got a very different structure now. Okay, and let's say it's got some hydrophobic residues on its surface now, exposed here, that were previously buried away in the center of the folded protein. Okay, right. Now, why does this make the misfolded protein sticky in quotation marks? Well, basically, this makes this protein more likely to aggregate, because if you imagine having another one of these misfolded proteins, and I might try to draw a mirror image one here, okay? This is probably destined to go wrong. Okay, like so. There we go, it's just sort of worked. Okay, so I've drawn another one of these misfolded proteins. All I've done is spun it round by 180 degrees, and it's got these hydrophobic residues here. What will tend to happen is, as I say, hydrophobic residues tend to get clustered together because water doesn't want to have to interact with them, so it forces them to interact with each other, okay, and it pushes them all together so that the contact between the water and the hydrophobic residues is minimized. So what tends to then happen is you get these misfolded proteins aggregating together. They're getting pushed together because they both have hydrophobic residues on their surface, and these hydrophobic residues get pushed together so that the water molecules don't have to interact with them. Okay, so that can drive misfolded proteins then to aggregate together. Okay, so there are 2 main types of aggregates that can form for misfolded proteins. Okay, one of them is driven by this sort of interaction that we're seeing here. Okay, the other is quite different. Okay, so the sort of aggregation that we have just looked at for misfolded proteins is what's known as amorphous aggregates. Okay, and amorphous aggregates literally the way that you would draw a picture for it is you would just draw blobs binding together. Okay, so basically, let's say this blob represents the misfolded protein. It's potentially going to have loads of hydrophobic residues now on its surface here. Okay, so this is going to represent our misfolded protein. These are representing our hydrophobic residues. What's then going to happen is you'll get another misfolded protein here, okay, and it will have hydrophobic residues. They will be forced to bind together through these hydrophobic interactions, as they're called, which really is all about water pushing all of the hydrophobic residues together. Okay, and then here's another one, and you'll eventually just form huge, great masses of these. I'll stop drawing the hydrophobic residues and just draw the main protein. And you'll just end up with large aggregates of them all bound together, and this is what's known as an amorphous aggregate. And as they say, it's driven by these hydrophobic interactions, but really what is meant by hydrophobic interactions is that water just does not want to interact with hydrophobic molecules, so it shoves all the hydrophobic molecules together so that it doesn't have to look at them, basically. Okay, right, and that's the driving force for these amorphous aggregates. However, there is another form of aggregation that misfolded proteins can undergo, which is quite common. We're going to see this a lot, okay, and this is the formation of what are known as amyloid fibrils, okay, and this is going to be the sort of aggregation that you get in many protein misfolding diseases. Okay, so amyloid fibrils. So I just want to start off by telling you what amyloid fibrils are now, and then it will help us out later on when we start to discuss the amyloidoses. Okay, so basically amyloid fibrils are formed from misfolded proteins, and the way they are formed is that the misfolded proteins produce a structure within them known as a beta hairpin. Okay, so this is the essential structure that underlies the formation of amyloid fibrils, a structure known as a beta hairpin. So let me just describe to you what a beta hairpin is. So a beta hairpin involves just two beta strands. Okay, so if I wanted to draw a picture for a beta hairpin, it would literally just look like this. This is my polypeptide represented as a line here, and you've got two beta strands here, one going in that direction and one going in that direction, so they're anti-parallel beta strands here that are interacting in the normal way that beta strands do. So it's a secondary structure, and therefore it's held together by these hydrogen bonds that form between the functional groups of the peptide links between the amino acids. Okay, so this is what's known as a beta hairpin. It's just two anti-parallel beta strands that are formed by the same strand here. Okay, so what tends to happen is these misfolded proteins can form beta hairpins, and then the beta hairpins can end up stacking on top of one another. So let me show you this. So here is one beta hairpin. Okay, and at present, the way I've drawn it is this beta strand here is interacting with this beta strand here. So the hydrogen bonds are in between these two strands. Okay, that's the way it currently is. But what you can do is you can stack another one of these on top here, then another one on top here. And what can happen is these beta hairpins can stop having the beta interactions between the beta strands that are in the same beta hairpin, and they can start having beta interactions with the beta strands that are below them in this stack. So what's going to start happening is, if I draw this picture again, here are these beta hairpins here. The interactions, the beta interactions, are now going to be between strands on one side. So this is going to start forming a beta pleated sheet on this side, okay, and then on the other one you're going to get a beta sheet formed here. Okay, so there's another beta sheet. And this can go on and on and on, and thousands of these things can stack on top of each other, making these two beta sheets basically, okay, and making a huge great fibre, okay, and it's called a fibril, which is just a tiny little fibre. So this is what's known as an amyloid fibril, okay. So amyloid fibrils then are formed when proteins misfold. They create these beta hairpin structures, and the beta hairpin structures can then stack on top of one another and form this huge great fibril in this way. Now, of course, the rest of the protein will be dangling off the side here. So this is only one little part of the protein. The rest of it can be dangling off the side here and can be folded however it wants, basically. And then, of course, that means that off the side of the amyloid fibril core, which is this stack of beta hairpins, you're going to have all the rest of these portions of protein stuck off the side. So amyloid fibrils aren't just the stack of the beta hairpin. That's the core of the amyloid fibril, as it's called. That's what's holding the entire thing together. But off the side of it, you've got the rest of each of these proteins that's involved in the formation of the core of the amyloid fibril sticking off the side. Okay, and that's a great big mess, basically. Okay, so that's another way that misfolded proteins can aggregate, by forming these beta hairpins and forming a massive great fibril like that. Okay, and it's actually incredibly common for proteins to aggregate and form amyloid fibrils, just as it's common for them to form amorphous aggregates. So those are the major two forms of aggregation that misfolded proteins can produce. Amorphous aggregates, because of the fact that we have this stickiness of the hydrophobic residues that are exposed, and these amyloid fibrils through the stacking of beta hairpins. Okay, right. Now, the problem then with proteins aggregating like this, forming amorphous aggregates and also the amyloid fibrils, is that once the misfolded protein has formed an aggregate, it's not then going to refold. Okay, so remember we said that misfolded proteins, if we were lucky, could return to their native state. Well, they're not going to return to the native state if they've aggregated together. Okay, because now, remember what the driving force for them to return to the native state was. The native state is actually the most stable state of a protein. It's energetically favourable for this to go to this, thermodynamically favourable. Okay, but once the proteins have aggregated together, they've got a new sort of thermodynamic stability there. So the proteins don't need to return to the native state anymore. Okay, to put this another way, this was unstable because of the hydrophobic residues exposed. But you know, if it's formed a great big amorphous aggregate, they're no longer exposed, basically. So now it's stabilized together, basically. Okay, so once you form these massive great aggregates, they're certainly not going to now refold spontaneously, basically, just by the laws of thermodynamics. Okay, you can, to an extent, use chaperones to try and pick apart aggregates and try and refold the proteins. Okay, and we'll see that a little bit later on. Okay, right. So the other thing I probably should say is that these aggregates, they can be intracellular or they can be extracellular, depending on whether the protein is intracellular or extracellular. If the protein is intracellularly and it misfolds in this way and forms an aggregate, then evidently the aggregate will be intracellularly. If it's an extracellular protein and it's misfolded in this way, then evidently the aggregate will be extracellularly. Okay, and again, we'll see examples of amyloidoses where the aggregates are intracellular and extracellular. Okay, right. So, just to complete then the proteostasis overview, okay, we've now seen how proteins are folded from nascent polypeptides to native proteins through the use of chaperones. We've seen the fact that they can misfold and hopefully be refolded. We've now seen the danger of proteins misfolding. We've seen that proteins misfolding can lead to them aggregating either by forming amorphous aggregates or by forming amyloid fibrils. What we now want to see is how does the cell deal with misfolded proteins that can't be refolded. Okay, well, the way the cell stops them aggregating into aggregates is that it has degradation pathways to destroy them. Okay, so misfolded proteins, if they can't be refolded, okay, by chaperones, they will go down degradation pathways. Okay, so again, we will discuss this in much more detail later on, but for now, let me just outline the two degradation pathways, the two major degradation pathways that cells have. Okay, so there is the ubiquitin proteasome pathway, okay, which we'll go into in a lot more detail later on. Okay, which is a way that proteins can be tagged, whoops, missed off a word, ubiquitin proteasome pathway, which is a way that proteins which have been misfolded can be tagged by having a ubiquitin molecule attached onto them, and then this sends them for degradation by a structure known as the proteasome. Okay, so the ubiquitin proteasome pathway involves them being tagged and then destroyed by this structure known as the proteasome. Okay, the other major degradation pathway is what's known as autophagy. Okay, and in autophagy, what happens is the proteins are sent for degradation in lysosomes, basically. So the proteins are sent to the lysosomes, and in the lysosomes they'll then be broken down. Okay, now autophagy, there are different kinds of autophagy, and we'll see a few of them later on. Okay, autophagy can be used to destroy single proteins like the ubiquitin proteasome pathway. Okay, or there are larger forms of autophagy which can be actually used to destroy whole protein aggregates which are formed within cells. Okay, so this is a way for cells to get rid of not just single misfolded proteins, but also massive great aggregates of misfolded proteins which are formed intracellularly. Okay, right, so the final word that I want to define in this video before we call it there for this video is the word proteostasis. Okay, now this word is motivated by the same sort of motivation as homeostasis. Okay, and what it means is the maintenance, that's what the stasis means, maintenance of the normal proteome. Okay, so what's meant by the proteome? The proteome means the collection of all the proteins within a cell. Okay, so this just means maintaining a normal collection of proteins within a cell that are functional. So maintaining a set of functional proteins that the cell needs for life, basically. That's what proteostasis is all about. And what we have just gone through is the crux of proteostasis. It's about synthesizing new proteins, making sure that they fold into the native functional three-dimensional conformation, okay, helping them to refold when they misfold, which is an inevitable process, and then if we can't refold them, then sending them to be degraded by either the ubiquitin proteasome pathway or autophagy, okay, and stopping proteins from misfolding and aggregating into either amorphous aggregates or amyloid fibrils. Okay, right, so that's all I'm going to say in this video. In the next video, what we will do is we'll discuss protein folding, this initial going from the nascent polypeptide to the native three-dimensional structure in a little bit more detail. Okay, we'll discuss what the difference between misfolding and denaturation is. Okay, we'll discuss the Anfinsen experiment and also the Leventhal paradox. Okay, so see you in the next video.